Hello, Dr. Metcalf. This is my provability assessment, and today I'm going to be discussing a little bit of my research that I've done at the University of Cincinnati in the last few years. My name is Joad Atway, and I'm a fourth-year double major in biology and neuroscience. So a little bit about my research is I conducted research on the RNase Q-Satvin. And an RNase is a molecule that cleaves RNA either specifically or non-specifically. So as you'll see here, Q-Satvin cleaves an RNA sequence every time it sees a cysteine. So these are the products you'll result. The interesting feature about Q-Satvin is it doesn't cleave between two consecutive cysteines, as you'll see here. It can cleave between cysteine and guanine, cysteine and uracil, and cysteine and adenosine. However, it doesn't cleave between two consecutive cysteines. My job was to figure out which amino acids are involved in giving Q-Satvin this characteristic cleavage property. I did this through site-directed mutagenesis. I made recombinants, so in the binding pocket of Q-Satvin, I made single point mutations, and I confirmed that the single point mutations were there with the BLAST alignment search tool. I would then culture the cells, purify the Q-Satvin protein with a histidine bound resin, and I would look at the gel to make sure it was the right size. This is some of the results I've gotten for the different mutants. And you'll see that the wild type has the highest activity out of all of the mutants. However, the mutants have more unspecificity binding. They're cleaving in other places that the wild type does not. That's the point of these peaks here. So through making many mutants of bacteria and expressing the Q-Satvin protein, I've been able to identify a few of the amino acids that play a role in giving Q-Satvin its characteristic cleavage specificity. My ongoing research will be testing to see if the mutants cleave with the same specificity with longer sequences like total tRNA. And I'm going to be doing some docking and some studies on how to get a three-dimensional structure of this with crystallography. Thank you.